Hello friends, welcome to EPG Pathshala. I am Dr. Imtiaz Hassan from Jamia Millia Islamia. Today we are going to talk on paper 10 biophysics, in which we will discuss about the module 20, that's the green fluorescent protein. In this module we are going to discuss about the discovery and historical milestone of green fluorescent protein. Then we will talk about the green fluorescent protein and its chromophore structure, especially with a special attention towards the environment. Then we will discuss about the mechanism of fluorescence of the green fluorescence protein. Then we will talk about some of the major applications such as the marker for gene expression, GFP in protein targeting, and as an active indicator of the cellular activity. And at last we will give you the summary. So let's talk about the green fluorescent protein. The elucidation of a structure and functional aspect of biological process within cellular network demands sensitive and selective detection method. So fluorescence has revolutionized this field of molecular biology with its immense application. Fluorescent marker and probes allow scientists to track molecular mobility of the cellular component, macromolecular interaction and conformational changes with a very high temporal and spatial resolution, both in a cultured cell and model organism. The use of various molecules facilitated the study of cellular processes under UV and visible regions. Green fluorescent labeling of biomolecules enables researcher to detect a specific component of a complex biological assembly even at a very lower concentration. So discovery and historical milestone of green fluorescent protein. We'll discuss about how it was discovered. So green fluorescent protein has existed for more than 160 million years in one species of jellyfish known as Aquaria victoria. The protein is found in photo organs of the aquaria of the jellyfish. Green fluorescent occurs when the aquaria protein interacts with the calcium ion and induced a blue light. Interestingly, this luminescent energy is then transferred to the green fluorescent protein and shifting of color emission towards the green side. Now we discuss about the photoluminescent properties of the green fluorescent protein. You can see here that the excitation spectrum of green fluorescent protein has a dominant maximum at 400 nanometer and a significantly smaller maxima at about 470 nanometer, while the emission spectrum has a sharp maxima about 505 nanometer and a shorter around 540 nanometer. So this is the spectral feature of the green fluorescent protein. You can see here in the spectrum that the spectral properties of green fluorescent family, in which the different colors are emerge at a different wavelength from 400 to 750 nanometer. Green fluorescent protein is composed of 238 amino acid residue and structure is highly unique and surrounding a central helix. Each monomer composed of a central alpha helix surrounded by an 11 stranded cylinder and anti-parallel beta sheets. Cylinder has a diameter about 30 angstrom and 40 angstrom long, and fluorophore is located at the center of the helix. You can see here the structure of green fluorescent protein, in which the beta strand surrounds a core in which the chromophore are located at the center. Green fluorescent protein can be used to target protein for and allow the expression of protein in a small sets of specific cells. Green fluorescence produced by green fluorescent protein is then allows the researcher to optically detect a specific type of cells both in vivo and in vitro. Here we will discuss about the chromophore structure of the green fluorescent protein. You can see here the chromophore structure and how the phenomena of fluorescence occurs in the green fluorescent protein. The active site residue of the green fluorescent protein are serine 65, tyrosine 66 and glycine 67. So you can see here that the deprotonated phenolate of tyrosine 66 is the real cause of the fluorescence, and Forster cycle leads to the fluorescence properties, and proton transfers from the histidine 148. You can see here in the structure that how the catalytic triad of green fluorescent protein are arranged in a three-dimensional space. Here we discuss about how the green fluorescence protein are exploited for the study of the gene expression and other biological activities. Green fluorescent protein can be placed as a tracer at the genetic location of protein coding gene, either as a fused recombinant protein or as a single entity driven by promoter of gene of interest. The inherent advantage of fluorescent proteins and fluorescent protein based biosensor for the study of cellular signaling process and protein localization coupled with the transcriptional activity have further motivated the development of plethora of new tools that is based on the green fluorescent protein. You can see here the different organism which having the fluorescence feature, green fluorescence feature only because of the genes which encoding GFP are transferred to their genome, and these protein are expressed and that produces the colorful organism. Here we are talking about how the green fluorescent protein genes are inserted into the vector, and then after insertion of this vector, this is transferred to the organism and that produces the green fluorescent protein. You can see here the structure of the PUG35 vector and PUG35P2B vector, in which the green fluorescent protein are inserted in the downstream site, and then it is used to produce the fluorescent organism, especially for to see the effect of particular gene in a test or model organism. Now we talk about the green fluorescent protein variants. Green fluorescent proteins are occur in different forms, different variants. Due to its potential for widespread use and evolving need of researcher, many mutant of green fluorescent protein have been engineered. Many colored green fluorescent protein mutant has been made, in particular blue fluorescent protein such as EBFP, EBFP2, azurite, M-calama-2-1, cyan fluorescent proteins such as ECFP, celurian, pipette, M-turquoise, and yellow fluorescent protein derivatives YFP, citrine, Venus, Vipette. BFP derivatives except M-calama contains either Y66H substitution. The critical mutation in cyan derivative is the Y66W substitution, which causes the chromophore to form with an indole rather than phenol component. Here we talk about the structural organization of green fluorescent protein variant. A broad range of fluorescent protein genetic variant of GFP have been developed over the past several years that feature fluorescence emission spectral profile spanning almost entire visible light spectrum. Extensive mutagenesis effort in the original jellyfish protein have resulted in new fluorescent probe that range in color from blue to yellow and are some of the most widely used in vivo reporter molecules in biological research. The primary motivation for developing blue fluorescent protein in the mid to late 90s was taken interest in creating matched pair of fluorescence resonance energy transfer experiments with a multicolor labeling option. You can see here the structure of chromophore motif of the green fluorescent protein variant like EBFP, the histidine 66, then glycine 67 and serine 65 triad are formed. However, in the case of EGFP, the tyrosine 66 in place of histidine 66 and threonine 65 and glycine 67 are there. On the other hand, ECFP, the tryptophan is located at the 66th position. However, tyrosine 203 is located in the EYFP. Now we talk about the spectroscopic properties of variants of green fluorescent protein. Different variants of green fluorescent protein were created with a specific mutation either with the centrally located chromophore or the main protein chain. Mutation facilitated the manipulation in the spectroscopic properties of green fluorescent protein, leading to their increased biological applications. The difference in the peak excitation of green fluorescent protein variant and the overlapping of excitation and emission range have led to their numerous biological roles. You can see here the emission spectra of different types of the green fluorescent protein variants. Fluorescence, excitation and emission spectra, solid and dashed line for typical members of 6 major class of green fluorescent mutants together with the chromophore structure believed to be responsible for the spectral characteristic feature. Spectra have been normalized to the maximum amplitude of 1 and actual green fluorescent protein are depicted as: for the A it is the wild type, B is the emerald, C is the H940, D is the topaz, E is the W1B and F is the P43. Now we discuss about the green fluorescent protein variants. The first is the wild type green fluorescent protein. The chromophore is an equilibrium between the phenol and phenolate form. It has two excitation peak at 395 nanometer and 475 nanometer. The second one is the phenolate ion in or EGF, in which serine 65 has been substituted with the alanine or glycine. It does not have 395 nanometer excitation peak. Third one is the neutral phenol, that is in which the mutation at 203 to isoleucine results the mutant that only has 399 nanometer excitation, presumably because of the threonine proton has no longer hydrogen bond with the phenol, thereby stabilizing it. The fourth is the phenolate ion with the stacked electronic system. Example is the Y fluorescent protein. Mutation of the threonine 203 to histidine, tryptophan, phenylalanine or tyrosine result in a yellow fluorescent color. The fifth one is the indole in chromophore CYP, cyan fluorescent protein, which have properties intermediate to those of BFP and EGFP. And the sixth one is the imidazole in the chromophore and phenyl in the chromophore, that is BFP, blue fluorescent protein, which have the excitation peak at the 383 nanometer. And seventh is the phenyl in chromophore. This mutant has the shortest excitation wavelength and no apparent usefulness. Green fluorescent protein, red fluorescent protein variants. Now we will talk about the red fluorescent protein variant. It is a novel fluorescent protein which was originally called DRFP583, but it is now commercially known as DS red. When the expression in the cell, it has fully matured DS red fluorescent protein and is optimally excited at 558 nanometer and has an emission maxima at 583 nanometer. The M-cherry chromophore is positioned near to the central axis of the beta barrel in a position of alpha helix that threads its way enough to center of the structure. DS red is an obligate tetramer with the tendency to form oligomer, which can lead to protein aggregation and interference with the localization of protein fusion in living cells. The inherent advantage of red shifted fluorescent protein and fluorescent protein based biosensor for the study of signaling process in neurons and other tissue have motivated the development of a plethora of new tools. Studying of subcellular localization using green fluorescent protein and the localization of a target protein could be detected using a variety of fluorescent protein fusion tags in subcellular compartments. eBFP is used for cytochrome C oxidase to the mitochondria. M-cerulean, a cyan fluorescent protein, and human beta actin highlights the filamentous actin cytoskeleton network. EGFP forming a cytoskeleton network when fused with the alpha tubulin. EYFP can be fused with the N-terminal 81 amino acids of human beta-1,4 galactosyl transferase to localize a yellow-green fluorescent marker to the Golgi apparatus. Focal adhesion become fluorescent with the chimera of M. Kucheria orange fluorescent protein MKO. The red fluorescent protein M-cherry highlights the nucleus and condensed chromosome when fused to the histone 2B in human cervical carcinoma cells. So these are some extensive application of red fluorescent protein. Now we discuss about the application of green fluorescent protein in the fluorescence microscopy to see the live imaging of the cells. The use of fluorescent protein as a minimally invasive tool for studying protein dynamics and function has stimulated by engineering of genetic variants with improved brightness, photostability and expression properties. Cells that express gene product tagged with the fluorescent protein can be imaged with low light intensities over many hours to provide useful information about the changes in the steady state distribution of protein over the time. You can see here that the EBF used for staining of the nucleus, then ECBF showing the mitochondrial dynamics, then EGFP showing the vinamentin, then the Golgi is shown by the EYFP, and DS-RED2FP used to see the endoplasmic reticulum dynamics, and HC-RED1FP used to see the nucleus. Here we discuss about the fluorescence technique in the live cell imaging. You can see the application of genetic encoded fluorescent protein has dramatically expanded the capability of living cell imaging in fluorescence microscopy by enabling investigator to target subcellular regions of interest with exact precision. In wide field fluorescence, the full aperture of emission light gathered by the microscope objective maximizes the record signal and simultaneously minimizes the required exposure time. Thus, a specimen can be imaged with a brief illumination period. You can see here some images that showing the live cell imaging using the green fluorescent protein variants. Here we discuss about the biological significance of the fluorescent tagging. Fluorescent tagging with the green fluorescent protein has a large number of applications and especially varieties of bioanalytical application such as in vivo imaging, high throughput screening of gene expression, diagnostic and proteomics, single biomolecular spectroscopy, fluorescence microscopy, flow cytometry, fluorescence resonance energy transfer, fluorescence in situ hybridization, and visualization of protein binding or the protein folding dynamics. It can also be used for tracking the biomolecules at significantly lower concentration in a living system. The non-destructive nature and the high sensitivity of fluorescence technique enables for a major other biological application. The requirement of a small measurements volume and low concentration of fluorescent material make it more useful and reliable technique for the development of reliable techniques. Change in absorption and emission wavelength values are always associated with the structural changes. So structure and dynamics of the cellular system can be monitored by the use of green fluorescent protein. Fluorescent labeling on biological molecules has been used to exploit their dynamics, kinetics and photophysical properties with a high sensitivity and specificity. In here we talk about the fluorescent protein labeling, especially the gene fusion approach. You can see here in the figure that protein-protein interaction can be detected by the FRET using two different green fluorescent analog. The two different proteins could be tagged with different fluorescent proteins, and their potential interaction could be detected by using FRET method. The detection of kinase activity using green fluorescent fusion protein, it has a wide application in the domain of insertion for the functional switching. Here we talk about the biological application in a special context of the protease sensitive FRET. You can see here that the first demonstration of FRET with green fluorescent protein come from a blue emitting green fluorescent mutant and a mutant GFP fusion via intervening protease sensitive spacer. The emission spectra of blue fluorescent peak at 447 nanometer, which fairly overlaps with the absorption spectra of mutant green fluorescent protein at 489 nanometer. The recombinant fusion protein would exhibit FRET, which could be disrupted using a specific protease to cleave a spacer, which would free the GFP in the cells. The appearance of green fluorescent would confirm the cleavage. However, the similar experiment with wild type GFP would not be satisfactory, since the excitation spectra of blue fluorescent protein is 383 nanometer, which could be also be directly and substantially excite GFP, having excitation peak of 395 nanometer even at in the absence of FRET. Using S65C or S65T along with the trypsin or enterokinase sensitive 25 residue linker region as a protease site, approximately 4.6 fold increase in blue to green fluorescent could be detected. The special advantage of green fluorescent protein based recombinant construct are the feasibility of incorporation of full length target protein, which could be expressed and assayed in real time inside the living cells or organism. Here we discuss about the FRET based small molecule sensor that are developed by using green fluorescent protein and red fluorescent protein. You can see here the concentration of a small molecule such as calcium or cyclic AMP can be measured by FRET based biosensor. Upon calcium release, CAM protein fused with the CFP interacts with the CAM binding domain of myosin light chain fused with the Y fluorescent protein, yellow fluorescent protein. Cellular signal leading to calcium release from the cells lead to the generation of a strong FRET signal. Upon excitation with 433 nanometer, FRET signal would show yellow fluorescent emission at 528 nanometer. That's how the FRET based a small molecule sensor can be developed by using CFP and YFP construct. Now we talk about the limitation of FRET between GFP to monitor the protein interaction. It has several advantages and disadvantages. The first advantage that works in vitro and in living mammalian cells, not just yeast. It can respond dramatically to post-translational modification. It has high temporal or spatial resolution in millisecond to microsecond. Interacting proteins can be anywhere in the cells and do not need to be sent to the nucleus. Degree of association can be quantitated if 0% and 100% binding can be established in situ. Efficiency of FRET at 100% complexation gives some structural information. However, there are some disadvantages. These such as the GFP must express fusion as a fusion protein in which the host protein and GFP must remain functional. If GFP are too far from each other, more than 80% or unluckily oriented, FRET will be failed. Even with no association, a spectral overlap contributes some signal in the FRET wavelength. Trace or rare interaction will be hard to detect. It needs negative and positive control also. That's the referent condition of the 0% and 100% association. And homodimerization is more difficult to monitor than the heterodimerization. Now we talk about the analysis of biological process through GFP tagging of target protein. It used as a reporter molecule to follow changes in gene expression over the time. Non-destructive and non-toxic tagging approach are implemented. It has a very stable structure resistant to the denaturation. Coding sequence can be cloned into variety of vectors, and GFP retains its fluorescence in cells from different species. GFP can be tracked in the living cells over time to time for studying developmental pathways. It can be directed to specific subcellular compartments. GFP coding region can be combined with the regulatory regions of another gene and observe changes in gene expression. It used to make a fusion protein for studying localization, turnover, and intracellular association of the native proteins. Now we discuss about the factors that affecting detectability of the GFP. The total amount of GFP, the number and copies of the gene, duration of expression, strength of transcriptional promoter and enhancer, and efficiency of translation. It including Kozak sequence and the codon uses. Absence of messenger RNA splicing, protein degradation and export, efficiency of post-translational fluorophore formation. Now we talk about the solubility versus formation of inclusion body, availability of the chaperone, hindrance to folding because of the unfortunate fusion of host proteins. Time, temperature, availability of oxygen, and intrinsic rate of the cyclization or oxidation affects the molecular properties of mature green fluorescent protein. Wavelength of excitation and emission, of course, affecting much, and the extinction coefficient and the fluorescent quantum yield, susceptibility to photos isomerization and bleaching, dimerization, competition with the noise and background signal, and autofluorescence of cells or the culture media at preferred wavelength are the factors that mainly responsible for the use of green fluorescent protein. Now we talk about the location of green fluorescent versus combined to a small sub-regions of the cellular or tissue, quality of excitation and emission filters and dichroic mirror, and sensitivity, noise, and dark current photo detector. So now let's summarize what we have discussed in this module. The major purpose of targeting protein for fluorescent probe is monitoring of biological process, subcellular localization, a specific detection of protein modifications, and isoform and reliable quantization of compounds. The increased use of genetically modified fluorescent proteins and advanced synthetic and natural fluorescent for live cells imaging has paved a way for wide spectrum of new optical approaches. Using latest technical advances, one can have a full complement or tool to visualize and record the measurements of cellular process for a large time scale with enhanced resolution. The attachment of a reporter group is required for the detection and biochemical cellular characterization of proteins and its binding partners, the visualization of cells and the cellular structure, as well as the tracking and the modulation of nucleic acid and protein in living system is mandatory to identify, map, and analyze the underlying biochemical process of cell signaling. We talk about some major application and the area where the green fluorescent protein are utilized. It is used for the environment and health science, For example, the pharmaceutical drug discovery, genetically modified organism in research, medicine, nutrition and bioremediation. It can be utilized in microbiology. Then the GFP can be used for the cell and molecular biology assay such as prokaryotic and eukaryotic cell structure and division can be analyzed, bacterial metabolism can be analyzed, and cell in a selective growth media can be analyzed. GFP can be used in the area of evolution for the antibiotic resistance genes, selection mechanism and adaptation to the environment. It can be used in genetics to see the DNA, RNA, protein and trait correlation, then genetic regulation of the transcription factors. It is widely used in the chemistry to see the protein structure, enzyme and catalysis, chemical properties of biological molecules, hydrophobicity of proteins and chromatographic separation techniques. And last, can be used for the scientific analysis such as uses of the chromatography to purify a protein, interpretation of experimental results and evaluation of hypothesis. With this, we end this module. Thank you very much.